down without thrusters the other night. Figure it spun out, but then on the way back up, I guess it just 6-8 does what it wants.
Getting a little spongier
Yeah.
with NA. More dead skeletons.
I think I'll change the
Promising.
speed to 0.3 on the way up.
Copy.
Bridge nav. Let's step 100 meters, 020, speed 0.3.
There's another sponge.
Nice.
See, there's more than one. Perfect.
When you guys are looking at the wraps, is it all visual, or is there a measurement for how many degrees you've turned back and forth?
There's a decimal from 0 to 1 for the.
That's based off the heading change, right?
Yeah.
Yeah.
So
0.1, 0.2, 0.3 sample.
And what would 0.1 mean?
1/10 of a turn.
Okay. And if you turn back, does it go back to 0?
Yeah.
Okay. Okay. Cool.
So if you turn 36 degrees, it'll be 0.1 revs.
But do they have the same starting point, the same direction as 0, or no?
When we zero them, we usually zero
on
our launch heading.
So you can zero it out. You can reset it.
Correct.
Cool.
That'll be at the end of your leash.
Yeah, sorry we haven't started moving yet. It's promising though. Sponge is out in the distance.
Yeah, it looks like on the backscatter, we're getting a stronger return as we climb, and then hopefully right around waypoint AB, we'll have some harder habitat.
Doing a heading change at the same time, so.
There's a nice sponge. That is a heterocony. Nice.
Zoom in there a bit, Steve, while we're waiting for the boat. I never checked the DSC. Is it
I don't think it's
set up?
running.
Remote desktop. Let's see. Do you know the way to get back into that?
Yeah.
So I just do connect here?
Yep.
Yeah.
And then, what is it?
Admin.
The morphology you get of these sponges down south is actually quite strikingly different than the ones further north, so they suspect there may be more than one species along the coast, although currently they're all designated as heterocony calyx.
Looks like it's capturing images.
It is capturing?
I think it says.
Yep, that's capturing. Not sure who's setting that up
Yeah,
now.
someone must have come in and set
Nicole.
that. I wasn't paying attention.
Yeah, Nicole came back here and set it up.
Yeah. Last time I checked, it wasn't running. So.
That sneak.
A little itty bitty ship in the bottom there.
Mm-hmm.
Okay,
They're
off
so
we
small.
go.
More amphiptyllum mixed into the sediment.
In theory, if we were to collect like a small heterocone, do you think it would survive getting collected and then being brought up to the surface so we could scan it for 3D imaging?
It will depend very much on the heterocone. So some of them are actually quite delicate, and like that one is likely to just kind of tear apart.
Okay.
One thing to check with will be Liz and Abby have a whole heterocone vase that was collected in 2016 from Grays Harbor Canyon.
Oh. Yeah,
And
it depends on how big it is.
Yeah, it's probably 18 inches across.
Oh, that's cool though.
Yeah, unfortunately, a lot of them, especially the ones down that we're seeing with the thinner walls and like the Pharea, once you touch them, they're actually much more brittle than they look, and they just start to fall apart.
Yeah. Yeah, there was well, they were in Olympic Coast. There were just so many like tiny heterocone sponges. There had been a recruitment event or something, and some of them looked like the miniature versions of the larger ones. And I was just in my mind, I was wondering if we were to collect them, I wonder how well they would hold up to then do a scan for later.
It's possible they do. There are a group that forms a fetus skeleton, but usually, as we've tried to just sample pieces of them, they are much more delicate than they appear. But if we find a good little candidate, we might be able to scoop it up. They are on our collection list.
Yeah, just something to think about.
Some tunicates.
And pick up the whole rock.
Yeah, good tunicates and a nice, those are dead heterocony they're growing on.
Oh, that would have been cool if they were alive. We made a good little 3D model, little pirouette.
Do you want to do that?
What's that?
It actually might be, the slope might be too high to get behind it, really.
Yeah, that's okay.
Yeah.
Oh, yeah, so there's a few here.
Getting a model of it.
Oh, yeah.
Yep, the slopes are much more interesting.
Interesting that we clearly have a mix of kind of this harder cobbles, but with soft sediment in between. We're still getting the sea pens. So unless we see one with a peduncle attached to a rock, it's got to be grandiform.
Mm-hmm. Look, yeah, so many dead heterocone.
The thing about the dead heterocone is it can be basically impossible to tell how long they've been there because they do basically turn into rocks. So it could be, you know, a year old or it could be 50 years old.
Go me to slow speed to 0.2.
Yeah. Oh, let me get
Bridge
out
now.
ahead.
Slow speed to 0.2 knots. Thank you.
Yeah, if you saw something like that little guy. Oh, sorry, what was that, Meredith?
I was saying we have a lot of these. We're seeing a lot of these that are all about the same size. Got a couple of Linnea on that sponge behind it.
I could just hold here.
Yeah.
If you want.
Yeah. I'm not
Bridge
sure what
now.
I want to do. Are we doing a model or are we moving on? Or?
Oh, no, we can keep going.
Stand by, Bridge. We're fine. Thank you.
And if we're comfortable, we can even start another transect unless, Meredith, you think there's too much collecting potential.
I think we can start another transect.
Okay, we'll have to get out ahead for that. So.
Okay.
Well, also,
whenever you're ready.
Right before we start, if we could trigger a Niskin. We haven't taken one yet.
sure.
Good thinking.
Why don't I hold here?
That way it will be.
Bridge now. Hold position.
Be paired to the transect then.
Thank you.
On the flatfish.
Yeah, it doesn't seem like a comfortable place to want to lie on the bottom with all these rocks. Several different kinds of sponges in view.
Gotten into a very spongy zone again. One of those bivalves. And yeah, still a lot of dead sponge. It seems like most of the sponges are growing on other sponges.
Mm-hmm. Do we know if the goiters on those heterocone sponges serve a certain purpose? Does it increase the surface area over which they can filter feed? Potentially?
That would be my suspicion.
Yeah.
See, they have
Okay.
kind of oscula openings there, so it would just provide more surface area. It might also change the way the water flows through the sponge.
Yeah.
Is there a specific place you wanted the Niskin triggered?
Just anywhere in here, kind of just so it's near where the transect is going to start, so that we can get just a sample that's paired to the transect.
Got it.
We're getting some little Paragorgia coral as well, little tiny guys.
Is it the first time we've seen the Paragorgia?
Yep.
Awesome.
Oh, there's a small cat shark.
Is that a small grenadier?
Yep. There's a big heterocone.
Yeah, you can see there in Argus view, the double cone.
Good morning, Tom.
That'll be a good one.
Tom just joined us in the science chat.
Yeah.
Oh, that's a cool one.
Super cool. You want to try to spin around this one and get some images?
Yeah. Yeah, if we're just holding.
Yep.
Might as well while we're here.
You want to pull up the DSC too?
I got it.
You want it?
You want to?
Yeah.
I zoomed in on Argus, but let me know if you need to take a look at the tether.
Yeah, we're good.
And then get the lasers off for it.
Yep. Coming on.
Yeah, there's lots of little recruits at the base. That's pretty cool.
Yeah, little guys. And a big one.
From the other angle, it almost looked like two that had fused together, but it just looks like maybe it's the same sponge. And are those smaller? The kind of like the gray looking sponge that looks like almost like tubes.
Yep, that's another sponge. That's a tube sponge.
Awesome shot, Steve.
Also got Phanea on there. That's just a Abby would love this shot. I think there's
Good.
5 or 6 different species of sponge there.
Dead sponge, a live sponge.
Zoom in just a bit, Steve. Thank you. You could zoom out on Argus there so I can see what's up there behind me.
Oh, that's a great shot in the Argus cam. Straight down. So there is this kind of like a divide in between the two. So it's two big separate barrels.
They're kind of growing together almost.
Yeah.
One of our scientists ashore, who's also one of the watch leads for the next watches, joined online, and we're filling him in as we go. Oh, we've got one of those porous plate sponge. Nice. Hopefully we'll be in some moldy cheese sponge too if we're seeing that one.
Yeah. Any insights on the moldy cheese sponge?
Not yet. It's a demo sponge. It's been a total pain to get DNA out of because we're trying to balance not like cutting up many pieces of it for DNA while preserving enough of it that we can possibly get taxonomy done on it. Hopefully we get some moldy cheese sponge today so that everyone can see what we've been referring to.
Yeah, I'm very curious. What do you mean when you say it's a demo sponge?
So there's 2 types of sponges we're seeing here. There's the glass sponges and then demo sponges. And so the glass sponges have the silica skeletons, some of which can turn confused. And then the demo sponges also use, they can use silica in their skeletons, but they tend to be softer without the rigid structure.
Okay. Like that spherical one we collected?
Yep, that's probably a demo sponge.
Okay. Come on. Where we're headed 020, where we?
020, and I think we can get 200 meters out of that. We'll go just past AB.
Oh, we're gonna do a Niskin.
Oh yeah. Forgot about that.
Yep.
Still dragging mud from the mud flat.
And do you all mind, since we were talking about starting a transect, I know we're very close to watch change. That's not going to cause any issues for you.
No, not for me. I get out of the chair and walk away. That's a big sponge.
Yeah.
Mm-hmm.
Come on, Herc. Hold still. Correct. Ah. Get a tight zoom there if you want, Steve. I'll get the manip out here. Let's look at the little squishy guys in there too.
Yep. There's also some encrusting sponge, and oh, there's definitely some dolomifera coral growing there.
Mm. The white one on the left.
Oh, and we do have a moldy cheese sponge right at the bottom of the frame.
Oh, that bluish
Oh,
looking,
yeah.
white looking.
That blue one, yeah.
Oops,
You'll
sorry.
get quite large, and they always have that blue color with like the black pores.
Yeah.
Interesting.
Is that all the way in there, is it?
Full zoom.
Yeah.
That's the first moldy cheese sponge of the dive of the expedition, and hopefully we'll see more.
Would that be too small to sample? Because that looks like it would at least tear away. Because sometimes we find them in hard rock.
That one, I'd be a little worried about knocking into these other more delicate heterocony just where it's positioned, but it's a good sign that it's around, and we should probably see more that'll be a little easier to get to than this one.
Okay.
Because they're also usually a pain to scrape.
Tom's asking if we should sample the stoloniferen.
I'm going to hold off here just because we are in front of this like really big, really delicate vase, and I don't want to do anything to mess it up, and just find some of these that are in a better position for sampling.
Okay, that sounds great.
And
I think we're ready.
Okay.
Oh,
Steve,
okay, good.
zoom out. Thanks.
So we'll get an eDNA snapshot of everything that's right here.
We're getting some high definition still, so the base of that sponge too. So.
Mm-hmm.
Yeah, that's a neat one.
Porch light.
It's on.
It's on.
Yep.
So this is our first Niskin.
Yep.
Yes.
Yep.
Must be with these snaggletooth jaws. Okay, Stevie on
Countdown.
the countdown.
3, 2, 1. Niskin.
I think it went.
Yeah.
I think it went.
What's the sample number there, Taylor?
That is sample number 064.
Roger.
can put the DSC a little closer there. Stick our stills camera in the sponge, I think.
Huh.
Tell me when it clicks.
Got it.
Did it get it?
Yep.
While the vehicle wasn't moving?
It was
Yeah.
a little shaky. But
Jake, you have control of the camera settings there?
it's right now it's taking interval shots every 30 seconds.
Yeah, but I see you have like ISO and aperture there.
Yeah.
Oh,
You
cool.
can mess with that and
And do you have control, full control?
yeah.
Cool.
You can, I think it's during interval shooting you can't
Oh,
mess
okay.
with the parameters, but
Okay.
when you come out of that, yeah, you can fully change any of the settings.
It's, what's the camera that's in there again?
I don't remember off the top of my head right now.
Super Razzmatazz $2,000 camera. $2,500 camera, I believe,
The
plus the lens.
Canon or a Nikon or something like that.
It's in that document that's right there on the desktop, which you probably can't get to.
No. I don't think
Can you toggle in and out of interval mode easily?
you can, but you have to start a new folder. It's a little it doesn't take too long, but if you
If
don't
we-
start a new folder, it'll
If we ever
start taking pictures and wipe the other ones.
come across something that really cool that we want, I'd love to jump in there and play with that a little bit.
Yeah, you can probably get on that monitor if you want to, or that
Oh.
computer.
I don't believe it actually does erase the files in the folder. It just says that it does. It might erase them off the memory card, but it doesn't erase them. I seem to remember him saying it doesn't really erase them, but we need to test that. So it would be nice to get in and out of auto shooting mode.
Yeah,
Let one of the shutter bugs play with it.
and for
Mosaic heteroconia to
mosaics
the
like
right.
that, it can
Sorry, what was that?
increase the capture rate.
Yeah.
Lasers.
Oh, lasers, yes.
Okay. Ready to rip here.
All right, so 020 speed.
Yeah.
Do you want to go 0.2 or 0.3?
Sure.
We'll go 0.3 and yeah.
Is the porch light still on?
Bridge nav.
Yeah.
Step 100 meters, bearing 020, speed 0.3. Thank you.
Is it off? It's not showing up.
Really?
So we'll mark this as the start, and then but we won't. I guess we'll see once the ship starts moving. We'll be able to.
No,
There
it's
it
off.
goes.
See,
Sometimes
is this
the delay.
is this T3?
Yes, T3.
Thank you.
Come on. Sorry. Okay. You come back down to the seabed here. That's why. Too many buttons. See if it'll do it. On the hill. Is it gonna crash? It's doing it.
Nice.
Oh, octopus.
Oh, nice.
So for Tom, we are now on transect.
We
We'll be
are
able
on
to
transit.
jump in and take over at the watch change. Got a lot of dead sponge still here. And wide sponge mixed in. Oh, we got some Berea now. Another glass
Oh, nice.
sponge.
Steve, are we
And I
full
think
out
some
on Argus?
Lego sponge.
Yep, pull wide on Argus.
Okay,
Yeah,
yeah.
I think I'm just
Double checking.
down on my delta a
I'll
little
double
bit.
check here. Yep.
It's almost like some of the basalt-looking rock like we were seeing yesterday.
You can put the sub bottom back up there if you want.
I'm going to copy and paste a quick screen grab of the backscatter that I have from OAT, just so it's in the dive plan so people have a better idea of what to expect.
Okay, and you have the targets overlaid on that?
Yes.
Great. Yeah, I might be able to quickly grab it downstairs and put it up here.
Yeah, I only, if you're interested, I was just going to put it up as an extra resource. I forgot that I had it.
Yeah. Great.
Might want to stop him up. Ready?
Roger.
Start going
Bridge
vertical.
nav. Hold position here.
Nice big plate.
So the steep part on my map is real, huh? What I was aiming for.
You nailed it.
Wow.
Little tiny heterocon sponges.
This is a nice, very vertical feature.
Yeah.
Mm-hmm.
Want to line up the sea prints for me?
Yep.
It is still kind of sedimented though, so I'm wondering, that's probably why we're not seeing too many corals.
Could be. I've seen in some areas where you like
Probably
sometimes
have
the
to
areas
close
that you
that
get a
little
lot of
dialog
sponges.
box first.
Oh, okay.
I think I'll start to move
You just
forward
don't
here. Is that
see
all right?
Yep.
Bridge nav.
Tilt up
that
a
many
bit.
corals.
Yep.
Mhm.
There have been
100
some,
meters, bearing 020, speed 0.3.
there have been some little little paragorgeous
Thank you.
twigs kind of mixed in.
Sponge garden.
Spine City.
Yeah.
Mm-hmm. Tom is noting in the science chat that this of a similar side of a feature nearby known as Piggy Bank is also filled with sponges. So it may just be something about the orientation to the local topography here that makes these conditions optimal for sponges.
Just gonna pause for a second here. We're starting to move the ship.
Okay.
Should be good to resume. Yeah.
Hot mic. Ouch. Yeah. Looks like we're ready to resume. Argus just started
My eardrum is ruptured now.
moving.
Good to watch, everyone. We're doing a change in video chair.
Yeah,
Thanks,
thanks,
Steve.
Steve.
Hey, Steve.
Thank you.
Dan wanted it.
That's Dan signing off. Dan and Jake signing off.
Yeah, thanks everyone.
Thanks. And I think with that, we will work on passing our SPL head logins over to Tom and Lisa, who are taking over from us.
Yep, it's been a pleasure. Thanks, everyone.
Yep. Thanks.
Yeah. Yeah, we're still going 0.3. Just let me know if you want to slow down.
Hey, this is Tom. I'm on now. Good morning, Lisa. It's morning. It's fine. It's just sitting in the dark all alone. Not even the cat's here to visit me. And there looks like an asbestos on that rock, and another one. We're starting to change habitat. We're still on transect.
Morning, folks.
Morning.
Good morning.
Good morning.
Audio check, 1, 2, 1, 2. Are we all here?
3, 4.
3, 4, 5, 6.
I'll be
Sorry, I'm late
7,
to
8
the
then.
party.
Mm.
Do we have our scientist online yet?
I hear a couple.
I'm here. This is Tom.
Hey, Tom.
Hey. So it looks like we have come about halfway through this transect. We'll just keep on this same bearing.
Roger.
That sounds great.
And the next
Yeah, so the general plan for today is basically we're just going to keep going up slope following the letter combos up.
sounds good.
And we're in Sponge City right now.
Looks it. Did they collect any of these sponges?
They collected a white ball sponge and one of the dead big yellow tan colored ones, but that's about it. We're actually getting different sponges now than they were getting earlier.
Oh, that's a cool one.
It's a heterocony with a crab family.
It's definitely larger than the others.
Yeah, there's a half dead one to the right. The bottom's dead and the top's alive still. Looks like an ice cream cone. They were seeing a lot of dead ones at the bottom.
Thanks,
yeah.
Kim. Standard box. Standard box.
Camera was looking up a bit, so you'd be running out of string.
Yeah, thank you.
Right there.
Thanks, Kim.
So do we have all the same people as last night?
I hope so.
Sounds like it.
Yeah, we better.
Somebody sleeping in.
Hopefully not. Do we have Lisa online?
All right, I guess we're doing introductions then. So this is Tom Leidig. I work at the National Marine Fisheries Service in Santa Cruz, California, and I study deep sea corals and fishes.
Yeah, and in the van, I'm Ben Gracian, sitting in the navigator seat.
Jess Sandoval on Herc.
Kim Moss on Argus.
I'm Marley in the video seat.
I'm Mary Duree in the data logger seat.
The entire science row. I'm going to keep these coming, Jess.
Yep. You just do 30 meters instead of
Roger.
100 or something like that, you know what I mean?
Oops.
Yeah, she's in the chat, but I guess she hasn't connected to VLink yet.
Yeah, I think, hey, it's Lisa,
If I
so
hear her, I
Lucy,
think.
can you hear me now? Yeah. Okay. Great, Lizzie. I think my FPL, I didn't have the talk function on, so now it's on.
Hi, Lisa.
Morning.
Hey, good morning. Lisa from, I guess Tom can hear me, maybe the others can't. So this is Lisa Loney. I work at the Office of National Marine Sanctuaries in the West Coast Regional Office. I'm the policy coordinator, and my office is in Monterey, California.
A lot of sedimented sponges here.
What was that question?
Oh, so many of these sponges seem sedimented.
Yeah, they're dying.
Yeah.
And that's one of the great questions of why some are dead and some aren't and some are half dead.
Right.
It's interesting. In 2010, we did the other side of this canyon, and it was also a sponge wall.
It looks like where Rennie Mark Sponge City was the steepest slope.
Yeah, but there's still a lot.
Yeah.
There's a dead one dead center now. Dead center.
How old do sponges usually live?
You know, that's a great question. I don't really know if people can answer that because I don't think they have continuous growth on their spicules. That's the hard part inside of them. So you'd have to find the oldest one of those. But the decomposed or the non-decomposed bodies like we see here to the left, they seem to stay around for a long time because we can go back and find lots of dead sponges in areas, and presumably the same one, for years. So their skeletons stay around for a long time even after they die. And they provide great structure for all these critters that are living in them. If Abby was here, or when she gets up, she probably could tell us more. And then you have the ones in the middle, these Rhabdocalyptus, which are white inside and brown outside. For whatever reason, all the detritus sticks to the outside, whereas most of the other ones, it doesn't.
So in their healthy form, they have that.
Yes. And you can tell by the inside is still white. Yep. It's probably one of the biggest groups that we don't understand. Because there's just so much diversity of growth form, even in the same species. You can see all these orange ones or orange yellow ones. They're called heteroconi. These ones with the holes in them look like waffles. They grow, and all of them, you can just see they all have kind of different shape. Sort of similar, but they grow differently. So it just makes it hard to tell if they're one species or two species.
So it's interesting that they have all these slightly different morphologies, but just seem to grow in the same areas right next to each other without any obvious.
Yep.
Yeah.
Well, it's interesting because these white Stercolyptus ones, these kind of vase-looking things to the left, they weren't down below. And we have a coral coming up on the right. Yay. The lone coral in the patch of sponges. But the Stercolyptus ones, you know, similar. They have lots of different morphologies and may be all the same species. It's just so tough to tell.
I see.
You have to collect them to be able to know. And we can't collect every one.
If you can tell the healthy sponges from the ones that are in trouble by the sediment, and they must all be seeing the same info, then the healthy ones must have some way to transport that sediment away.
Yeah, it could be that, or it could be something is killing the other ones and it's not. The healthy ones are healthy, but they're different parts of it. So the parts that aren't healthy could be something's eating it. It could have some kind of parasite or some critter that is attacking it. That's Abby is kind of working on, or Abby and her colleagues are kind of working on some of those things. Because there are just so many questions on, like, why are we in a healthy patch now when there's a dead one in the middle? What killed that one?
So it actually looks like we've come 205 meters from the target. That's,
All right,
yeah.
then I guess we can end transect.
Sweet.
Roger that.
And this is T3.
All right, and then so what's the next? Where's what next waypoint on here? AC over there.
AC to the northwest.
So yeah, let's just kind of mosey over toward AC.
Great. So it looks like we'll be coming over this local maximum, Jess, and then it's kind of downslope on the north side of that.
Okay.
So
Before we move, can we zoom in on that rock to the left of the lasers with the white things on the top of it? The little white yellowy spots?
right to the bottom left of the laser.
Just almost, well, for me, it's, yeah, just slightly bottom left.
Bottom left. Sure.
So just, oh, never mind. I see what it is, a dead sponge. Never mind.
Oh, Roger.
I was thinking it was sometimes in your eye. Yeah, let's zoom in on the dead sponge anyway. We'll take a look at it.
Sure.
It's right in front.
Is that a yellow sponge overgrowing it, or is that remnants of the?
All right, Marley,
That's
go
a
ahead.
great question.
Get there.
It looks like it has a few little live sections at the top, barely. The last little remnant of its life.
Hold on, buddy.
All right, go ahead and come wide, Marley. So it's going to be our
315 would just be directly there. It seems.
Do you want us to, Science, do you want us to hop, kind of go straight up slope and then go along the contours? That might give us the best visibility.
Sure,
Or
that
we
sounds
can
good.
go right straight there.
Yeah, it's just there's a little dip here, so either way.
Yeah, Whatever you guys feel best. It's kind of similar to last night. They're just pre-picked points. There's nothing special about them. So.
Do you want to go more directly upslope, Jess?
We can. That works out all right. Then we can kind of go along this contour and not lose too much delta.
Yeah, Roger. Just might be a little pitch here, so let's just go right up it. So let's do 280.
280, Roger.
And we hit the mud now.
Sorry,
too, so.
2350.
350.
350.
350, Roger. 350, okay.
Well, we stopped the transect at the right time, that's for sure. Now it's back to mud. But I see more rock coming. Lots of different sponges.
Biggest one.
So to the left, no, he's not too happy. To the left of the lasers, there's these tube sponges. They grow up in tubes and also below the lasers. Just all these different growth forms of sponges.
Oh, yeah.
But the tube ones still have one big chamber
Riley,
on
you want to push
the
in,
bottom.
please?
Yeah, they start from the same point.
Oh, that's cool. I've never seen that. All right, go ahead and come wide. Thanks, Marley. You can do a full wide, and I'll just get squared up on my porch here.
Yeah. Okay.
Huh.
So on the last watch, when I started watching about 340, they found a moldy Swiss cheese sponge.
All right.
Which is
go ahead and push back. Oh.
It's an elusive sponge that's pretty small. So look for anything that looks kind of white and blue on the bottom, probably less than 10 centimeters. So it'd be some little tiny thing stuck on the bottom.
White.
White and blue?
Yeah, it's actually
like Roquefort cheese?
Yes, well, we call it the moldy Swiss cheese, but you could call it blue cheese too.
Okay. Because when I heard moldy cheese, I thought, well, that could be any color. Could be purple too. Okay. Moldy Swiss.
We've never really been able to sample them because they grow low on the rocks. And if we found a little rock that had it on